for you. Okay, so now what we need to talk about in this particular class is all about your cell membranes. Okay, so we're going to talk about the cell membranes and how these cell membranes are going to help you in the process of transport. Now, if you see in our previous sessions, okay, what we have done is we have gone over the structure of your cell membrane, right? We spoke about how exactly your cell membrane is going to look and what are the different ways in which the cell membrane is permeable to different molecules. Okay, before we go back and revise all of the things, okay, I would ask you to note down all the difficult words in your notebook, and then when you look at the back of your textbook, okay, what you will see is the definition which is written in bold for you. Okay, so wherever you see that there is a definition, just note down that particular word, and then later as a practice for you, what you will do is you will note down the entire definition from your textbook. Okay, so that you can also have a small revision and you would also have some notes written down for you. So let's talk about your cell membrane. Now, previously we have discussed that this is typically what your cell membrane is going to look like, right? It is going to have these two lipid heads adjoining like this. Okay, they are going to be placed in this manner, and then there are going to be certain molecules which are going to be placed either all the way through your membrane, or they're just going to be lurking on the surface of the membrane, and these are going to be either your protein channels, or these are going to be your carrier proteins, or these are just going to be certain groups, okay, certain prosthetic groups that are going to be associated with your you can say cell membrane. Okay, and they are usually used in the process of identification, reception, or even just communication with each other. So cells are going to use these molecules to communicate with each other, right? So we've been over this particular part. We're going to go back to those basics again, and we're going to try to learn how exactly your membranes are going to conform themselves. Okay, what are the different shapes and configurations that your cells are going to go into because of the presence of a cell membrane. Okay, we are also going to learn that cell membranes don't necessarily always need to encapsulate another cellular material. Okay, it is not always necessary that when you talk about membranes, these membranes be present as a part of a cell. Sometimes these membranes could be formed. Okay, so you can have this same structure formed without the involvement of these protein structures. And what these lipid molecules, okay, what this lipid bilayer would do is it would just be present so that it could encapsulate other material than your cellular matrix. So it does not always mean when you talk about a membrane, you're talking about a cell membrane. There are multiple other membranes that we will look into, right? So this makes sense. Can we go ahead now? Yeah.
Yeah.
Okay, so now let's look at what are the different properties that our membranes are going to have. So we discussed that you are going to have your membrane, which is going to be semi-permeable. Right now, when I say semi-permeable, what I mean is there are going to be certain substances. I'm writing the entire sentence. Okay, there are going to be certain substances that are going to be given entry inside the cell, and then there are going to be other substances that are probably not allowed to go inside. So because there is this sort of distinction, okay, or because there is this sort of separation as to which molecules are allowed to get in and which molecules aren't allowed to get in, we will say that your membrane is going to allow only certain things to cross through. And that gives it its characteristic property, which is that it is a semi-permeable or semi-permeable. The permeability is limited only to certain particles. Now, when I say that there is a permeability limited to certain particles, it also tells us that our membrane, okay, this membrane that we have is not only semi-permeable, it is also selectively permeable. I didn't have that. Okay, so you're going to have these substances that are not only going to move inside your cell, but your cell selectively chose to allow these materials to move through or not move through the boundary, right? So this brings us to two very important properties that your membranes are going to possess, one being semi-permeability and the second one being selective permeability. Selective permeability. Right? So these are two most important factors that we need to consider every single time when we talk about our membranes. Okay. Now, when we talk about these membranes, let's just draw a very simple structure of the membrane. So what I'm going to do is I'm just going to bring in the cell membrane structure for us. Okay. So I'm going to talk cell membrane, and then we're going to copy this image, and then we're going to use it for understanding everything that I am just talking about. Okay, just going to take a few longer minutes because I have a bit of difficulty in terms of internet today. Okay, so please just bear with me. Now, while we do that, okay, while this image is being downloaded, I'll talk about what exactly we need to look into. Okay, so now when you talk about the substances moving through your membrane, we have already seen that the molecules can move through the membrane either without bribing your membrane, or they have to bribe the membrane with ATP molecules so that that particular molecule is allowed to move inside the membrane. And based on this particular kind of activity, we said that your cells, okay, or you can say that your transport, okay, the cellular transport that takes place inside the cell is sort of kind of divided into two major categories. Okay, these two categories being active transport and passive transport, which means that you are going to have, okay, you're going to have a type of transport where you are going to require a expenditure of ATP. You are going to require to put in certain amount of ATP. Okay, and then there is going to be another kind of transport where you are not using any amount of ATP. When you are doing any transport in a cell, okay, whenever you're carrying out any kind of transport, for some reason my screen sharing just stopped. Okay, now whenever you're carrying out any particular kind of transport, if this transport happens to be against the gradient, okay, against the concentration. Now, what I mean by against the concentration is there is a certain number of molecules present outside my cell. This I don't think is going to work. So I'm just going to go ahead and draw this thing for us. Okay, so let's say this is how our cell membrane here is at this point. Okay, there are certain molecules in a higher concentration on the other side. Okay, so I'm going to say that this is my cellular matrix and this is my extracellular matrix. Cellular matrix is everything that is contained within the cell membrane that belongs to a cell. Extracellular matrix is all the substances inside the body of an organism, but not an entirely part of the cellular structures. Okay, so this could be any other molecule that allows your cell to carry out its function, but does not always necessarily have to be inside a cellular matrix. So that is going to be your extracellular matrix. Now, in your extracellular matrix, you would notice what you have is a lower concentration of a particular substance, and inside you would notice you have a higher concentration of the substance. Now, if the transport was taking place from cellular matrix to extracellular matrix, we would say that this is along the concentration gradient, because you are having the movement of the particles from the higher concentration to the lower concentration. That is along the concentration gradient. In this particular kind of transport, you would not expend any amount of ATP, because this is just happening because there is a higher and lower concentration gradient that has been created. But if you have the similar kind of situation, but the transport is happening in the reverse order, which means that from the ECM, it is coming to CM, that is from extracellular matrix, your substance seems to be moving towards the cellular matrix, then what you are going to say is that this is a particular amount of, or you would say that this is the kind of your transport that seems to be happening against the concentration gradient. And when this kind of transport is happening against the concentration gradient, here you are going to spend some amount of ATP in getting this particular task done. Okay, so when it goes in yellow manner, okay, when it is moving from higher concentration to a lower concentration, we are going to call this kind of transport as passive transport. Okay, I'm just going to put this. So this is going to be your passive transport, and the other way around is going to be your active transport, right? So you have these two modes in which the transport is going to take place. Now, along with this, okay, what we also have already seen is that when these particles are going to move through, okay, whenever you're going to see that, okay, there are certain particles that are present inside my membrane, and these membranes are then going to allow these particles to move. Okay, so I'm just going to make a very simple diagram here for us. So let's say this is what I have here with me. Okay, I'm going to make a protein channel here, and I'm going to make a protein channel right here. Okay, so the lighter part is the internal part. What I have done is I have cut my channel into half like this. So what you're seeing is a transverse section of it. And here I have another protein, and let's just simply color it. Okay, so this is what I have with me right now. Obviously, all of these are going to be my lipid heads this way. Okay,
I have
and
a question.
yes, tell me.
So is it that the inside part will always have like a higher concentration and the outside something like that?
No, so that is the reason why you are going to have this transport take place. Okay, so the main reason, or you can say the primary reason why these molecules are constantly moving, why there is a dynamic flux in their concentrations, is so that not all the time, not in all the instances, your one side is having higher concentration. This is the reason why substances keep moving from one side to the other, so that you do not come across a particular situation where only one side seems to have a higher concentration and the other side is always suffering with lower concentration. This has to be avoided, because if that is going to be a part, okay, where you come to a point where the concentrations are not changing, you would say that your cell has reached its you would say maximum threshold to stand for a particular concentration. Okay, and the cells who are no longer exchanging material are going to be killed off. Okay, because a cell has to be always in a dynamic state of flux, just like your electrons need to compulsorily keep on moving in the orbits. If they stop, they are going to come to an inert stage, and that is where your atoms are going to no longer react, right? A similar thing is going to happen with your cells as well, and your cells are not going to be functional anymore, and that is where the cell death is going to take place. Okay, and once the cell is dead, even then the substances do continue to move from one side to the other, because the cell is going to be degenerated. Okay, it is going to be degraded, and all the substances from the previous cell are then going to be extracted. The useful substances are going to be used just as they are, and the rest of the material is going to be broken down into its constituent parts and then utilized by the body, because that is how our body works. Okay, so all the substances that you are going to have inside you are still going to be used up even when the cells give up on their particular function. Okay, so this is something that you need to remember at all times. Now in between here, okay, in this particular structure, you already know that there are going to be these cholesterol chains, right? And this is basically how your cell membrane is going to look like. Now the movement of the molecules, okay, or the movement of the particles is basically going to be in a way, okay, that you have certain channels. So let's say this is our protein channel. Okay, a protein channel is going to have certain substances on one side, and there are going to be the same substances on the other side, but with a difference in their gradient, difference in their concentration. Okay, and what these substances need to do is they need to come to this particular protein channel, and then they need to pass through this protein channel and come on the other side. Okay, sometimes this movement can happen in the opposite direction. If it is happening from a lower concentration to higher concentration, we said it is going to be your passive transport. If it is if it is happening from the higher concentration to lower concentration, that would be your passive transport. If it is happening from lower to higher, it is going to be your active transport. Okay, now you have your protein channels. Okay, the protein channels are specifically going to take in your molecules, okay, and they are going to aid the movement of these particular substances, and then they are going to take your substance from one side to the other, and they're just not interested in why this substance is coming in or going out. Okay, they just keep on moving these substances from one side to the other. But then you are going to have another kind of transport, okay, where you might need a substance to come and associate itself with your protein channel. And when that substance, it doesn't always have to be ATP. Okay, let me just put that out here for you. The substance that is going to be utilized to make the movement of other substances easier inside the cell membrane does not always have to be your ATP. It can be any other substance that can work as a conformation change molecule. Okay, the term I'm using is conformation. That means we're talking about the shape and the size of a particular molecule. Okay, so when I say the conformation change is going to take place, it means that I had this particular shape molecule with me. Okay, and as soon as this particular substance came and associated itself with my molecule, there was a conformation change, and this molecule now looks like this, and the associated molecule releases itself. As soon as the molecule releases itself, my substance reverts back to his original shape. This is how my conformation change works. Sometimes the conformation change is elastic, or you can say plastic, which means that it can form and then re-go back to the shape it was. Sometimes the conformation change is more permanent, and once a molecule has changed its shape, it will possess that shape till it finishes its function. Okay, so it will remain in that particular conformation and not change it. Right? That makes sense. Okay, you followed that
What
part.
substance that attaches?
These substances would be any of your enzymes. These could be your hormones at times, or these could be your saccharides as well. Okay, depending on what is going to catalyze this function, you're going to have molecules come and associate. Right, so you can refer to these as your subsidiary molecules, or you could say that these are just my channel activators. Okay, so you can refer to them for right now for your understanding as the channel activator proteins. Okay, now once we understand this, okay, we need to see why are these molecules, coming and going toward your cell, or what are the different ways in which the conformation change can take place. Okay, so you could have your membrane go into several different kinds of conformations. One conformation that we usually see is where you're simply going to have a arrangement of your molecules this way. Okay, so I'm just gonna make it like this. Okay, now let's say this is an arrangement of your molecules right now. There is another molecule underneath here. Okay, so there are two lipid heads. Okay, one is the upper layer. Let's say the gray is the upper layer and black is the lower layer. Okay, and the conformation that your molecules are present in right now is like this elastic band, right? So they're basically like this. This is a one layer structure, and there is a front side and there is a back side. Okay, so this is the conformation that my membrane is in right now. Now you can see, just like the membrane, okay, because this is a ring structure, this can bend and twist in any manner so that it can occupy smaller space. So I can just bend it and twist it into two turns, and then I can arrange it into a smaller place. I can again twist it, and I could bend it this way so that it becomes a little more condensed, right? This is a property that we are able to see with your membranes. And one of the most common examples where you have heard of this happening is your mitochondria, right? So you have always learned that a mitochondria is a dual layered structure, and the inner layer happens to be larger than the outer layer, right? You've always learned that. And then you also learned that the inner layer, as it is a larger layer, as it is a little longer, what it has done is it has undergone this conformation change, where instead of being a spherical or just like a ring structure, it has gone into these convolutions like this. Okay, and it has gone under these convolutions so that it could be placed into a smaller structure, right? So this is something that your membranes are able to do, and this kind of flexibility is offered to your membranes by these cholesterol molecules that you have. Okay, so this would be the cross section. Let's say what we saw over here right now is the cross section of a particular membrane that could be involved in making a sphere. Okay, so you could have a sphere like this. It is completely full. Okay, and what I'm gonna do is I'm gonna make those yeah, so let's just do it this way. Okay, so there are all these membrane molecules. Okay, there are all these lipid molecules here, and what you have done is you have just gone and cut the membrane this way. Okay, and now what you're looking at is the cross section, right? So I'm just gonna mention over here, this is the cross section, right? This makes sense. Are you able to understand? So I took a cell, okay, I took a membrane, and what I have done is, I'll give you an example here. So I have this conformation in which my cell membrane is. What I do is, if I cut this glass from here, and then if I look at its top surface, this is what I'm able to see. Okay, this part is what I'm able to see. Now, it means that your membranes are also going into a spherical kind of a conformation where they're present as a three-dimensional structure. Now, I'm not sure if you remember this part, but we have also gone over this. Okay, so let's say I have this particular structure here. Okay, this is a sphere, and no matter what side or what location you look at it from, all the sides, this is what you're able to see. These each individual blobs that you see are nothing but your lipid heads. And then that means that inside, okay, either there are there's like an entire new layer inside. So we'll just make another new layer inside here. Okay, let's make it with a different color. Okay, so let's say that there is this other layer all the way inside, which is completing the structure, making it look something like this. So there is upper layer this way, and there is a lower layer of lipids this way, right? And then this is a dual layered sphere that has been created, which we normally see. Okay, this is your normal cell membrane. But if this kind of internal part is not present, okay, if you have just one layer of lipid with its tails pointing inward, and then they contain some kind of substance inside, usually a fatty substance which cannot be transported all by itself. Okay, such a structure that is present over here is called as a missile. Okay, so I'm just gonna write it over here, missile. And multiple are called as miscellaneous. Okay, so these are the molecules, okay, or these are the, you can say, structures which are made up of lipids. Okay, they have a hydrophobic tail, and they have a hydrophobic and hydrophilic head and a hydrophobic tail, and then they are going to contain some kind of substance in them, and because they're spherical, they've been given a specific name which is called as a missile. Okay, and multiple is called as miscellaneous. Now this is one way in which you could observe your cell membrane. Now if you like, I don't know, I never did pay attention in mathematics, but in case you ever did, okay, your math teacher might have taught you this particular concept where they might have told you, okay, this is a circle, and if you take any one point on the circle, okay, let's choose this, and if you drew a line that goes and touches just one point like this, a tangent, okay, this is called as a tangent. If you draw this particular line, you would see that a circle at some point is straight. Okay, that means if you were to make numerous lines like this touching one point on your circle, you would see that the entire surface of a circle is actually made up of tiny lines. Okay, that is creepy. I mean, who went through and learned all of this? But apparently this is what we were taught in school. Okay, so if this is to be considered true, if the Euclidean geometry is to be understood, okay, this is what Euclid actually said in the first place. Okay, and then all the we just started following what Euclid said, because apparently he was a very smart guy. Okay, so this is what we have learned, and if this is to be considered true, if what Euclid has said is to be taken as north, okay, you would say that at some point, okay, because your lipid molecules are really small, there is one point on your circular membrane or on your spherical membrane where the membrane is unlike a sphere and is in the form of a sheet. Okay, so this is something that you would observe. So I'm going to just place this here. Okay, and you will see what I'm talking about in a minute. So it is present like this, and then you're going to have something that looks like this. Okay, so this is something where you're going to see that this membrane is in fact nothing but a sheet like this. Okay, this is where we observe that your membrane can take up another conformation where it can be present as a sheet. Okay, not just present as a circle or as a bent structure or as a miscellaneous, but also in the form of a sheet. Okay, there is also another way in which this membrane can occur. Instead of being circular like this, it could go into a sandwich form like this. Okay, I don't know why I call this a sandwich structure. I guess it just reminds me of how your sandwiches look. So it looks something like this. Okay, if you were to add cheese and tomato, it makes sense. Again, maybe some patties. That's not just a sandwich, it could be a burger also, but this is the kind of shape that your membranes can also go into. So this way and here. Okay, and what you would notice in this particular structure would be that these tails are just going to hang out like this. Okay, and then these are going to sort of join here. Okay, now please remember this kind of U-pin structure that we are talking about, because this similar concept where there is bending over onto the same structure, right? This is the same membrane that has simply just bent over itself. We are going to learn something similar to this in your DNA as well. Okay, so we are going to see that not only your cell membrane, but even your DNA likes to get into this particular kind of conformation where it is just bending over its entire own surface so that it could make this particular kind of a shape. Okay, so this is something that we see in our membranes understanding. Okay, we also have understood that whenever you talk about these membranes, okay, whenever you talk about the cell membrane and when you talk about its structure, we saw that there are these glycoproteins, there are phospholipid molecules, and then there are these transport proteins, and you have just normal proteins associated, you have glycolipids, you have carbohydrates that are going to make up some part of your glycoprotein. Okay, all of these substances are basically present so that some kind of transport could take place in your body, right? So now what I'm going to do is I'm going to save this part. I'm going to take you to a different app where we are going to learn a simple concept. Okay, so I'm just going to open this. I'm just going to take this here. Please ignore everything that you see on the screen. It's just some random things that have been drawn. Okay, so I am going to take my common brush. Just give me one small second. Okay, so now let's say this is my base membrane. Okay, this is how my membrane looks like. Okay, I'm just gonna start this. All right, here we go. Now in this membrane, what I'm gonna have are these different molecules. Out of those different molecules, this little guy is of importance to us. Okay, so we take this, we make this into a background, and these are the molecules that we are majorly concerned about. Okay, now what you're going to be noticing is that there are certain substances who might want to try. Sorry.
Are you drawing something?
Yes, is it not visible? Okay, just give me a second. For some reason it did not share. Is it visible now? Are you able to see my screen now?
Yes.
Yeah. Okay. So now what we are going to look at is that, let's say there is this particular molecule which is present here. Okay, this way. Then I'm going to slowly move it upward. Okay, now once this molecule is seen, okay, what you're going to notice is that as soon as this molecule starts to move towards it, okay, towards this particular protein, what this is going to do is it is going to kind of change its orientation a little bit so that this molecule moves a little inside. Okay, once this molecule has come in over here, okay, this will try to revert back to its own shape and then open in the other direction so that this molecule can actually go in, and then this will come back to its original shape like this. Okay, so we need to add that molecule here. Just give me 1 second. So it is here, then it is here, then it comes here. Maybe another one moves in. Okay, and then what you're going to be noticing is that this guy again shuts off. Okay, so give me 1 second. I'll just make it a little slower. So this is basically what you're going to be seeing, which is going to happen in your cell membrane. Okay, this is the kind of movement that you're going to see whenever you talk about your channel proteins transferring some kind of information from one side to the other. Okay, now this was happening all by itself. It was happening because there was a higher and lower concentration of the molecules, and it was just allowing your material to move inside. Now let's say if this entire thing had to happen with involvement of ATP, okay, then what would happen is the ATP molecule would come along with these substances. Okay, they would come here, and then what you would observe is that your protein molecule, okay, this entire molecule would in fact undergo a conformation change. Okay, so let's make a conformation change here. It would now look something like this. Why, you would ask? It could be just so that there is a ease in opening and closing of your molecules or the channel proteins, or this could also happen just so that a particular kind of molecule can pass through. Okay, so if I'm going to make another example out of this for you, okay, I could say that this is also happening probably because again, this is your cell membrane. Okay, and what you have here is a particular kind of molecule. Okay, so I'm just going to make this molecule here, this molecule here, this molecule here, and it seems to be moving forward. Okay, and then all the way through your membrane. Okay, now the channel protein that you have has a very irregular shape. So this is the way that your channel protein is actually like. Okay, and it has a lumen that will allow only this much or this size substance to move through. And obviously you can see that the molecule that you have is a little larger. Okay, so what you would do is you would attach with this molecule another substance that can bring about the conformation change. Okay, now once we see that there is a molecule that can bring about a conformation change, what you may observe is that the lumen sort of opens up like this, and that is also a conformation change, because now what has happened is that your molecule looked a certain way and now it doesn't look like that. Okay, so this is the kind of conformation change that you may observe. So this molecule has not yet come around, so it will not have a conformation change. So this will go through, this will also go through, and this will also make its way through here. And then your substances or your protein channels will come back to their original shape and size. Okay, so this is what we need to understand. Okay, so this is here, there is a particle associated here, and then this particle is no longer a part. Okay, and it will just go away. So we'll play this part. Oops. Make it a little slower so that you may follow through. Okay, so this is something that you are going to see occurring inside your membranes as and when the particles try to move in and move out. Okay, now if this particular kind of movement, okay, is happening for your solid particles, you would say that this is nothing but your diffusion. Okay, but if this kind of movement is happening for your water molecules, you would say that this is your osmosis, right? So this is something that you have already understood even in your previous grades, and you already know that this is what your membranes are going to do. Okay, this is how they are going to transfer different substances from one side to the other. Just give me 1 second, I'm just going to take one sip. Okay, so tell me, till this part where we spoke about the substances being moving from one side to the other, are you following me? Is this something that is making sense?
Yeah.
Right, so now, okay, now let's say this is the same membrane that we have with us. Okay, now what you would observe happening with this membrane is somewhere along here. Okay, let's say there is this particular kind of an antibody that is associated. Okay, so let me just put this antibody on the main diagram here. Okay, so let's say I have this antibody that is associated. Now, if we consider this to be an antibody, then the only function that we know this guy is going to do is that whenever there is going to be an antigen entering in the system. Okay, so let's say that there is a anonymous antigen here somewhere like this. Okay, and it seems to be moving from wherever it is coming. Okay, and then this particular antibody, okay, will kind of associate with that particular antigen, and then it will kill that antigen. Okay, so you would notice that along with the cell membrane playing a part in transferring the substances from one side to the other, this particular membrane is also playing a part in keeping the cell membrane and the cell safe. Okay, so this is something that you would see. Okay, now this molecule that we just drew here, okay, is actually nothing but your this is nothing but your glycoprotein. Okay, so this is your glycoprotein. We're just gonna color this up and we'll give it the name. Coloring with a crayon is a little difficult. I'm gonna give it its name. This is glycoprotein, right? And now what my glycoprotein is doing, it is kind of coming and catching my particular antigen, and it is inhibiting the activity of that antigen. Similar to this, okay, you can also have another molecule here. Okay, so let's say I have a particular protein this way. Let's color it up. Okay, and this particular protein, okay, that I have has another cholesterol or maybe a carbohydrate molecule associated with it this way. Okay, so it has this carbohydrate chain here. Okay, and all that this particular guy is doing is through the entire day. Okay, so we'll just bring it here. It just looks into different directions on the cell. Okay, so let's just get rid of this part. Yeah. So it is going to look in one direction, and then later it is going to look in other direction. Why is this part coming on my main picture? Okay, here. Okay, so it's gonna look here, and then it's gonna look here, and then again it's gonna look here, and again it's gonna look here, and then if it sees something, okay, which could be of value, it's gonna be like, hey, you have a message. Okay, and then this message is going to be either transferred inside the cell, okay, or this message could be transferred outside the cell. Okay, so here what this particular molecule is seen doing is that it is simply transferring the message from one side to the other. Okay, and this transfer of message is to be carried out so that your cell is always updated. Okay, not just the internal part of the cell, but also the external part of the cell needs to keep itself updated as to what is exactly happening outside and inside. And this particular process is called as cell signaling. Okay, so you have these molecules that are either sending the signal in or they're sending the signal out. And in this particular manner, they are maintaining this particular kind of information transfer. Okay, so this is something that you are going to see. Now when you talk about this particular kind of signaling, okay, this kind of signaling is going to have a very different kind of pathway, and it is going to have a very different mechanism in which it is going to work. Okay, so we're just going to make a small loopy here so
Is
that
there also glycoprotein?
that could be a glycoprotein. Your cell signaling molecule could also be just another just another substance. It could also be another antibody. So the nature of your cell signaling molecules could differ from cell to cell. Okay, so in plants, animals, fungi, protists, and bacteria, the cell signaling molecules are going to be very very different. Okay, but the mechanism or the pathway remains conserved throughout. Okay, so you may have different ways in which the signal is accepted and given back, but the mechanism remains the same. Okay. So
Yeah.
I need to put this on the main diagram again. So here is our main diagram. Let's put the color to it. Okay. All right. So now what we need to see is that there is a certain kind of stimulus that you're going to receive. Okay, so I'm going to say at this point of time, what you're receiving, okay, your cell is, this molecule is still just looking here and there, right? So this is going to be the stimulus part. Once the stimulus has been received, your receptor, okay, is going to get activated. What is the receptor? This black guy is your receptor. Okay, this receptor is an important part. Okay, so we're going to put this block over here. What this receptor is then going to do, this receptor is going to transmit the message or signal. Okay, so we are going to write over here transmission of message. Right now, once this transmission of message has taken place, the message is going to reach a particular structure or a particular part, which is called as the target. Okay, sometimes this is also referred to as the effector location. Oh, we shouldn't have put everything on the same page. Just give me a second. Target, which is also called as effector. And then what you're going to see is that there is a sort of movement, okay, or some kind of activity is going to take place. So we'll put this here and then we will write. Okay, so this is how your cells are going to work with a certain kind of stimulus. Okay, so this is again something that you need to follow through. If you go to your textbook, okay, the same kind of pathway is shown to you. It's just in a little bit more complicated manner that it is described, but the signaling molecules usually use a very easy transport system. They get a stimulus, they take it to a receptor molecule, the receptor molecule will simply transmit the signal to an effector or a target molecule, and a response will be generated, similar to what you learned in your reflex action, right? So similar to that, you're going to see something happening over here. Okay, now whenever this particular kind of structure, or whenever this kind of signaling is something you study, you will see that although this distance through which the message has to be transmitted is small, okay, the mechanism of your cell signaling is still going to be in the manner that your nucleus and your brain does it. Okay, so your brain is also going to have this kind of signaling that it takes place, where it is going to have a receptor molecule and an effector molecule, and thereby a receptor organ and a receptor effector organ. Okay, the similar kind of conversion of your chemical impulse into your electrical impulse, and then the electrical impulse then again getting converted to the chemical impulse, and then chemical again getting converted into electrical. Similar to that, even though this message has to be transferred in a very small space, that kind of conversion may also be included in your cell signaling. Okay, and the most effective way in which we can say, or we can just kind of prove evidence that this kind of cell signaling does occur, is by taking into consideration the drugs that we inhale or consume, or the medicine that we consume, or it could also be with kind of your odors that you smell. Okay, so you know that the odor was smelled by this side of your nose. That kind of signal goes through your brain, and then the receptor molecules of your nose, the olfactory molecules, get activated. And the same process that happens from your receptor molecule to the brain also happens in the olfactory organs. And the same way your cells come to know, oh, there is some kind of an odor, and maybe we either need to stimulate the cilia so that you can sneeze. Okay, or maybe we need to just constrict our nose down so that we can inhale less of that particular odor. So all of this happens not only at the cellular level, but it also happens at the subcellular level. Okay, so each and every single molecule of your cell membrane is also going to participate in this entire process. Okay, so it's very, very deep, and it is going to be very, very instantaneous. But while it is instantaneous, it also requires a lot of tiny guys coming together and doing their job efficiently so that this could actually be a thing. Okay, so this is something that we have to look into more detail when we go ahead in this chapter. What we are going to do post this understanding is we are going to understand um, does the shape and the size of a molecule that is going to move in and out of the cell, excuse me, so I probably smell something. Okay, so does the shape and size of a molecule that is moving inside the cell or maybe moving outside the cell have a particular kind of effect on how the transport is going to take place? We are also going to look at whether the pressure or the surface tension of the molecules has a role to play in how they get transported from one side to the other. And if it does, then are those molecules also undergoing the process of reconfirmation, or maybe in the process where their conformation changes, or does the cell itself is the only structure that undergoes changes in its conformation? So that is something that we need to look at. And once we understand that, we will look into one of the most important mechanisms, okay, that your cell probably undergoes, which is the process of taking in substance by will. Right now all of this is happening because a cell kind of notices a gradient difference. But what if you had one singular cell who had a will to allow certain substances to come in or let the substances go out? So would a cell which is maybe a unicellular structure be able to control its cell membrane so that it could carry out all these processes by will? Is something that we are going to explore in our next session. Okay, so I want you to go through your textbook, read all this material, and then when we come back, the first question that we will address is how diffusion takes place if osmosis is going to take place, how diffusion takes place if diffusion is going to take place in a particular manner, what are the different things that are going to affect it, and then we are going to look at one single cell and we are going to understand if its will is going to affect all of these. Okay, so is there anything that maybe you did not understand? There's something that you want more description on or something, anything at this point? Or do you just want to look at the recording one more time and then you want to come back if you have any doubts?
I'll have to look at the recording once more.
All
I
right, so
think I did get everything, but like if I like zoned out a little in the middle,
that
then I'll have to look at it.
okay.